Our next speaker is Godfrey Pearlson. Dr. Pearlson is the director of the Olin Psychiatric Research Center, which is up in Hartford, affiliated with the Institute of Living there as well as with the Yale Department of Psychiatry. And Dr. Pearlson has done some really exciting work trying to understand how the labels that we put on psychiatric illnesses, such as bipolar, schizoaffective disorder, schizophrenia, may not actually be carving nature at its joints. We need to gather lots of data, brain and other data, in order to better understand how different individuals with psychiatric disorders may differ from one another and may respond to treatment differently, independent of the labels that we currently use in our limited state of knowledge at this time. So I'm looking forward to seeing what he has to say about that. Thank you, Dr. Pearlson, for being with us today.
Hey, good morning. Okay. I've only been here 2 seconds and I've already screwed things up. That's great. Beat my personal best.
We need to play.
All right, thank you. Okay, so good morning. I'd like to thank Dr. Crystal and the organizers for inviting me. This conference is always very interesting, and I learn a lot, mainly through talking to people in the audience. So how did I get interested in the topic of psychotic and non-psychotic bipolar disorder to begin with? Well, my mother was the youngest of 14 children, and 2 of her sisters had quite severe forms of psychotic bipolar disorder. One of my aunts who was affected lived a few hundred yards down the road, so we would go visit frequently, and you were never sure whether you'd meet a perfectly ordinary housewife who worked in her husband's tailoring store, or someone who was out in the garden screaming at the neighbors and singing to her flowers. And these illnesses would happen periodically, and it fascinated me that someone could be so sick when they were ill and then return to being a completely outwardly just average person when they were well. So that sort of piqued my interest. And what I want to talk about today is just how bipolar illness can present in these rather different forms of psychotic and non-psychotic illnesses, which I'll define for you. And just explore whether biology can tell us anything about whether the psychotic form of bipolar illness is just a more severe form of the same illness, or it's really a different beast, as it were. And that's something that we can probe using neuroimaging and genetics. And I'll tell you what we've learned on that journey. So clinical presentation, like what sort of symptoms people have, how old they are when they first get sick, what the longitudinal course of the illness is like, are the sort of information that we use in DSM-5. And if we don't know anything about the underlying biology of these illnesses, then that clinical presentation is the obvious place to start. And this is where clinical medicine began. So if you had a chronic cough with a fever, then that's a syndrome, and that's something that a physician can make note of. But before the onset of clinical tests like chest x-rays and being able to culture what people coughed up in a petri dish, or to put a probe down into someone's lung and take a sample, then all you know is that your patient has a chronic cough and a fever. And if you treat someone for tuberculosis when they have lung cancer or vice versa, you're not really helping your patient other than if you hit lucky. And psychiatry is gradually catching up with this in terms of understanding the underlying biological causes of psychiatric disorders. So at least symptoms get you off the starting block. So psychiatrists can agree among themselves if someone has certain symptoms or if they don't. So psychiatrists can talk to each other, and if they're talking about a form of bipolar illness or schizophrenia, they're generally in agreement how the person is presenting clinically. But this reliability is really bought at the expense of validity. We're not sure what the underlying causes are, so our classifications are not really valid in the sense that scientists would understand that. So what we have is really appearances, how patients are appearing, but not the underlying essence, what's going on under the hood. So why do we care? We care because unless you can figure out the underlying biology, as Dr. Crystal said with cancer, you can't really understand how to individualize treatment for the person that you're seeing and their biological illness. So just to bring us sort of focus in on psychotic and non-psychotic bipolar disorder, about 40% of bipolar patients have a psychotic set of symptoms during an episode of mania or depression. And psychotic symptoms are hallucinations and delusions. So hallucinations are realistic sensory phenomena that the person actually believes in, but that are generated inside of their brain rather than by anything in the outside world. And delusions are fixed false beliefs that are culturally inconsistent, that other people don't believe, that it's impossible to argue them out of, although these are irrational and other people would not support them. And the main question that we asked, although it's a very simple question on the surface, is just, is there anything different biologically between individuals with bipolar disorder who don't or do have psychotic symptoms? So the sort of biological measures that we used to classify them were functional MRI, where subjects rested inside of an MRI scanner that Dr. Sinha described to you, just quietly for 7 minutes. We didn't give them any kind of task. And we've used PET scans to measure the neurotransmitter dopamine and its receptors. So what are the hypotheses here? Well, one is that non-psychotic and psychotic bipolar disorder sort by mood. So they're completely or partially overlapping, but they kind of hang together. And they're separate from individuals who have no psychiatric illness, just healthy controls, and from individuals who have schizophrenia. Second set of hypotheses is that because patients with schizophrenia also have hallucinations and delusions, they overlap with individuals who have psychotic bipolar disorder, and that non-psychotic bipolar disorder patients would overlap more with healthy controls. Or perhaps healthy controls would be in a completely separate category and non-psychotic bipolars would be in yet a different category. So really what I'm talking to you about this morning is just sorting this stuff out. So what are the symptoms in individuals who have psychotic bipolar disorder? So many of them have delusions, about 70%, and most of those delusions are grandiose. And about 40% of patients with psychotic bipolar disorder have hallucinations. Most of those hallucinations are auditory or visual. So typical non-delusional ideas that people with mania have are just grandiose ideas. So these are all examples I've got recently from patients who showed up in emergency departments. So one person said, "I can sing as well as Beyonce." So that's possible, unlikely but possible. So I asked for a demonstration. And all I can say is that this person will never headline at Coachella. A second patient told me, "I bet I could run for governor of Connecticut and win." So again, it's not a delusion. At least 50 other people believe that. And all I have to say about that is be very careful what you wish for. And in depression, typical non-delusional ideas in bipolar disorder and in major depression are self-blaming. And typical quotes here that I've heard recently are, "My whole life has been a disaster," which is possible, and "I've been a bad mother to my children," which is also possible. Although in both of those cases, that was actually demonstrably not true. So typical bipolar delusions are, again, in mania, grandiose delusions. "I'm queen of the universe." That was clearly not true. And "I alone hold the key to peace in the Middle East." Although Jared Kushner may believe that implicitly, that is not a delusion, but in this case, it was in this person's case. And in depression, "I'm already dead and was the worst person who ever lived. And if I think bad thoughts, then millions of people will die because of me." Again, self-blaming delusional ideas that are typical of major depression or the depressive phase of bipolar disorder. So Gabrielle Carlson, who at the time that she wrote this paper in the 90s was in the intramural program at the National Institutes of Health, described what she called a third stage of mania. And she said, patients who have this are showing up mainly with bizarre behavior, hallucinations and delusions that are absolutely not consistent with their mood, so that they might be manic, but they do not have grandiose delusions. They're more likely to be very paranoid. And rather than being clearly very euphoric or very depressed, they're much more likely to be dysphoric or mixed. So she said, despite symptoms that might have otherwise prompted the diagnosis of schizophrenia, that is, mood incongruent hallucinations and delusions, these patients appeared clearly manic both earlier in their course, when they were ramping up to this severe stage, and later as the episode was resolving, when they were coming down from this period of psychotic bipolar illness. So examples that Gabrielle Carlson gave was, patient was frightened, talking and crying constantly, pacing, I'll never get out, I have cat's eyes, he crawls around inside of me, and so on. And please let me die, I can't take it anymore, I'm at the National Institutes of Hell. And they were at the National Institutes of Health, but this is what we call the National Institutes of Health when they don't fund our proposals. And following treatment, the patient reverted to a typical manic state. So the obvious question that we want to pose first is, are patients with any of these forms of psychotic bipolar illness just manifesting plain, ordinary bipolar illness in just a more severe form? So this is a paper that came out in the last year in bipolar disorder, and from any current level of functioning or background level of functioning that you look at, just in terms of histories of substance abuse, if they're rapid cyclers, how functionally impaired they are, or how well they do in the outside world, patients with psychotic and non-psychotic bipolar disorder look practically identical. So this is really good evidence in a fairly large series that the two sorts of bipolar disorder are not more and less severe forms of exactly the same thing. They appear to be as similar as two different illnesses could be. So a second level of approach has to do with the underlying genetics. And Jimmy Potash, who's currently the chairman of psychiatry at Johns Hopkins, looked at this back in the early 2000s. And he noticed that psychotic bipolar illness tends to breed true, as it were, within families. So if someone in your family has psychotic bipolar illness and someone else in the family has bipolar illness, it's likely to be of the psychotic form, and vice versa, as it was in my mother's family. So he thought that psychotic bipolar disorder may identify a subtype of value that's distinctly genetically different and biologically different. And the hope of genetics has been to identify underlying risk genes for neuropsychiatric disorders such as schizophrenia and bipolar disorder, and lead us to the underlying neurobiology that would then identify rational treatments. And the person who's done the most research in the genetics of psychotic and non-psychotic bipolar disorder is a Canadian researcher called Jim Kennedy. And I couldn't find a picture of him online, so this is a picture of a Jim and a Kennedy, just to kind of give you the idea. And what Jim Kennedy's reported is there's a kind of confusing overlap between gene risk variants for schizophrenia and bipolar disorder, and if anything, the genes for psychotic bipolar disorder more resemble risk genes for schizophrenia, although, to be fair, the jury's still out on that. It's really too early to answer that question. And something else that also tends to muddy the waters is what geneticists call assortative mating. So this is a picture of a patient's night out in the 1930s at the Institute of Living. And people will meet who have similar symptoms. So person A with schizophrenia meets person B who has psychotic bipolar disorder, the conversation would, we imagine, go something like, "Oh, you have hallucinations too. Cool. Netflix and chill?" So these individuals are attracted to each other because they share similar symptoms. So we can imagine that they would get together, and we can imagine that their offspring share a mixture of psychotic and mood symptoms, and that this is perhaps where psychotic disorder, bipolar disorder, comes from and muddies the water as far as tracing the genetic basis of these disorders. And Elliot Gershon, who's one of our collaborators, has looked at this and has produced some fairly compelling arguments for why that's not the case and that these really are separate illnesses. So PET scanning. This is something we did when I was at Johns Hopkins a long time ago. You can use a cyclotron, a charged particle accelerator, to make carbon-11, which is an isotope of ordinary carbon that breaks down very quickly. It has a 20-minute half-life, and as it decays, it emits two photons at 180 degrees that can be picked up as radiation in a PET scanner. So what you can do is incorporate the carbon-11 carbon dioxide that comes off the charged particle accelerator, the cyclotron, into a drug that is very much like Haldol, called spiroperidol. It's used as an antipsychotic drug in Japan. And if you look at the brains of individuals with psychotic and non-psychotic bipolar disorder and with schizophrenia compared to healthy controls, then interestingly, the number of dopamine D2 receptors in schizophrenia and in psychotic bipolar patients are significantly increased and not different from each other, whereas the number of dopamine D2 receptors in non-psychotic bipolars look no different than those of healthy control subjects, and again are very, very similar in their number. So we're seeing a sort of biological separation already, psychotic and non-psychotic bipolar disorder, at least based on the number of dopamine receptors. And recently, Oliver Howes in England, who's actually been a grand round speaker here at Yale at least a couple of times, performed a similar PET experiment in the last year where he looked at dopamine synthesis in bipolar disorder and schizophrenia, again in psychotic and non-psychotic bipolar disorders. And what he showed in the basal ganglia here deep in the brain, an area that Dr. Sinha just described to you, is that the dopamine synthesis capacity in the psychotic bipolar disorder group looked very similar to that of patients with schizophrenia, and that of the non-psychotic bipolar disorder group looked very similar to that of healthy controls. And when he plotted within the psychotic bipolar sample the severity of psychotic symptoms in schizophrenia, shown by these little black triangles, and psychotic bipolar disorder, shown by these blue dots, against dopamine synthesis capacity, is that it was a straight line and a very significant correlation. So the more dopamine your brain was making in the basal ganglia, the more psychotic you were, which speaks a little bit to the underlying mechanism again, and again distinguishes between psychotic and non-psychotic bipolar patients. They look biologically different. So what I wanted to talk about next is the resting state paradigm in functional MRI. So in an active task, the person is asked, and this person's actually lying on their back in a scanner while they're doing this task, is find the letter T from a bunch of other stimuli in the background on a computer that's in front of your face in the scanner. So they're thinking, where's the letter T? And so that's what's in their mind. In the passive task, you just have the person look at a cross on a screen and relax. And they're told, think about nothing in particular, just sort of chill out in there. So the average person's thinking, my toes itchy, when am I getting out of here? It's my wife's birthday, I better get her a card or I'll be in big trouble. Got to buy kitty litter on the way home. Just random thoughts. And interestingly, what that identifies is what's called the brain's default mode. So this is a very well-known, well-described circuit that appears in all of us if we just relax and think about nothing in particular. So this appears in healthy controls who are relaxing. It's still there when people are asleep. It's still there when people are in a coma. And it's still there under anesthesia, even in non-human primates. So the same circuit appears in monkeys. This is something that's very well preserved throughout the mammalian kingdom. There are even small equivalents in rats and mice that you can identify. And the questions that we can ask about the default mode are, what are the connections within regions of this single component? So this is marked with a little asterisk to get you to remember it, but you can think of the default mode or any of these brain circuits as just a bunch of people or brain regions, chatting with each other on their cell phones. So I've labeled a couple of these regions 1 and 2, and this is region 1, this is region 2, and there are all these other regions. And all the time these regions are chattering with each other when you're just relaxed. Next thing that you can think of is connections among the brain's components. So again, this is the default mode over here with the asterisk, but the default mode is actually talking to your motor circuits, to circuits that are related to concentration and attention, or circuits that are related to future planning. So the default mode is not only chatting with itself, but also talking to all of these other circuits. And that's just if you smush together everything that's happening during those few minutes in the scanner and look at it all as one thing averaged together. But of course, when you're averaging things together, what you're missing is what's happening from second to second. So the other way that you can look at connections among these components is to look at how the components change across time. One way of thinking about these circuits is just as a sort of game of whack-a-mole. So one circuit pops up, like the default mode, and then you're thinking about planning something in the future, so the default mode pops down and one of these other circuits pops up. And that's really how the brain works. These things are constantly evolving in a landscape of communication among brain circuits. So we've done a couple of experiments with this. One is a study called PARDIP that's looking at non-psychotic bipolar individuals that we compared to other individuals that we had in our archive from a related study called BSNP. This is something that Alan Antichevich in the Department of Psychiatry here looked at a few years ago with us. And what he found is that parts of the frontal cortex during resting state in schizophrenia and in psychotic bipolar disorder, shown over here, were on the negative side of 0 and very similar to each other, whereas patients with non-psychotic bipolar disorder and healthy controls, shown over here, were on the opposite side of 0 and hung together. So it's yet another suggestion that psychotic and non-psychotic bipolar disorders are very different from each other, and that the non-psychotic form of bipolar disorder is closer to, although not entirely overlapping with that of healthy controls, and that the psychotic bipolar disorder patients over here are similar to, although again, not completely overlapping with patients with schizophrenia. So the BSNP study that I just alluded to is 5 sites, one of which is here in Connecticut. And in BSNP 1, we looked at 2,500 individuals with schizophrenia, psychotic affective disorder, psychotic bipolar disorder, and schizoaffective disorder, and their first-degree relatives, unaffected relatives. And what we found is that across a huge variety, like 50 different biological markers that were derived from EEG, IQ tests, eye movements, and functional MRI, and other things, that psychotic disorder patients, psychotic bipolar disorder patients, schizophrenia patients, and schizoaffective disorder patients were remarkably similar to each other. So the obvious question is, well, what about non-psychotic bipolar disorder? So we did a separate study called PARDIP that studied 150 individuals with non-psychotic bipolar I disorder and compared them to the BSNP patients. So this is something that we're still working on, so I'll show you data that are not published and have not yet been presented in 877 subjects from BSNP1 and PARDIP. So there are 170 patients with schizophrenia, 203 with psychotic bipolar disorder, 71 with non-psychotic bipolar disorder, 310 healthy controls, and 123 with schizoaffective disorder. And when we looked across time, what the brain was doing in 30-second windows across the entire time resting in the scanner, thinking of nothing in particular, we pulled out 3 states that the brain went into. So state 1 was highly connected. A lot of cortical regions were chattering with each other during this time, some subcortical regions. State 2, where red is much more active and more correlated and blue is less correlated, State 2 was positive and negative and not as highly connected as State 1. And State 3 was what we call just sparsely connected or really not very connected at all, where these brain regions are not talking with each other much and the brain just seems to be kind of spacing out somewhere. And what we wanted to find is if these different diagnostic groups spent different amounts of time in states 1, 2, and 3, and how frequently they change from one state to the other. So what we found is that healthy controls in bipolar subjects with psychosis differed from each other a lot, as did healthy controls and patients with schizophrenia, but that patients with non-psychotic bipolar disorder look no different than did healthy control subjects. We couldn't distinguish them accurately from each other. So here are some of the metrics in the controls, the non-psychotic and psychotic bipolar patients. So the dark blues are the controls, the reds are the non-psychotic bipolars, and these are the psychotic bipolars. So in state 1, 2, and 3, you can see that patients with non-psychotic bipolar disorder and healthy controls, the dwell times look very similar, whereas patients with schizophrenia and psychotic bipolar disorder look very similar to each other. And that was also true for the amount of time, the relative amount of time or fractional time that patients with these disorders spent in states 1, 2, and 3. So the fractional time as opposed to the dwell time was similar in psychotic bipolar disorder patients and schizophrenia patients, and in healthy controls and non-psychotic bipolar disorders, as well as the number of state transitions. And just to blow this up a little bit so you can see it more clearly, healthy controls and non-psychotic bipolars were very similar. Psychotic bipolars, schizoaffectives, and schizophrenia patients were also less and very similar. So the overall conclusion is back to those models that these disorders sort by psychosis, that schizophrenia and psychotic bipolar disorder are more similar to each other. Healthy controls and non-psychotic bipolars are also different, similar to each other. So these basically bipolar with and without psychosis are discrete disease entities that pair up differently and have different biologies. So the take-home messages, long story short, about 40% of bipolar patients have a psychotic form of the illness, manifested by delusions and or hallucinations, that psychotic bipolar disorder and non-psychotic bipolar disorder seem to breed true in families, that psychotic bipolar disorder is certainly not just a more severe form of the disorder. It looks clinically and functionally very similar, but it's biologically very different, and that excess dopamine may be involved, as it is in schizophrenia, in psychotic bipolar disorder. And the two forms of bipolar disorder we think are genetically and biologically different and require different treatments. So I'd like to thank everyone in our group, particularly Shashwath Mehta and Alan Antichevich, who've done a lot of the analyses I showed you together, and all of the research assistants, some of whom are here today, who collected all of these data in BSNP and PARDIP. And maybe we have time for a question or two. Thank you.